Assalamu alaikum warahmatullahi ta'ala wabarakatuh. In this third lecture of amino acid metabolism, we are going to discuss biosynthesis of non-essential amino acids. Our objectives today are to define non-essential amino acids and explain how they differ from essential amino acids, list the precursor molecules that are used in the biosynthesis of non-essential amino acids, to describe key biosynthetic pathways, and to highlight key enzymes and cofactors, and finally discuss relevant biomedical issues. Regarding biosynthesis of non-essential amino acids, it is important to remember that they have short synthetic pathways. And when we say essential and non-essential, we mean that in a nutritional context. That's to say, all 20 basic 20 amino acids are biologically significant and important. When we say non-essential, we don't mean that it is not important. All of them are important. Missing one amino acid, particularly if it is relevant to certain structure or function, is going to affect the body negatively. So essentiality here actually refers to the ability of the body to synthesize or not synthesize that amino acid. And there is survival advantage in the sense that the body retains the ability to synthesize the easy amino acids that have short synthetic pathways, while losing the ability to synthesize the difficult amino acids that have long or complicated pathways. So the body will take those from outside, from the diet. Let's see here in this slide. This is a list of essential amino acids: arginine, histidine, etc. To synthesize these amino acids, the body needs up to 59 enzymes. However, to synthesize the non-essential amino acids such as alanine, aspartate, asparagine, etc., the body only needs collectively a total of 17 enzymes. You can see the difference. So the body, from a developmental point of view or from an economic point of view, decided to retain the ability to synthesize those that only require 17 enzymes, and to take those in the diet, and to let go or to let some other organisms synthesize those for it. Biosynthesis of non-essential amino acids can be from amphipolic intermediates. For example, alanine from pyruvate, asparagine from aspartate from oxaloacetate, glutamate from alpha-ketoglutarate, our key enzymes, or modification of an existing amino acid. So the body can use an amino acid to synthesize another amino acid, for example, cysteine from methionine and serine, glycine from serine, and so forth. And the process of biosynthesis can be, for example, through transamination. We have 4 amino acids that are synthesized through transamination. Namely, they are alanine, aspartate, glutamine, and serine. Each one of those is synthesized through transamination. The body can also synthesize amino acids through assimilation of free ammonia. So it can take up free ammonia and incorporate it to synthesize either asparagine or glutamate. And the third way of biosynthesis of non-essential amino acid is modification of an existing amino acid, and this applies for cysteine, glycine, proline, and tyrosine. We have already discussed transamination, so we'll go quickly through it. Transamination is transfer of amino group from one amino acid to an alpha-keto acid analog of another amino acid. The alpha-keto acid will form the new amino acid, and the amino acid that donated the amino group will be converted to an alpha-keto acid. So it is a reversible reaction, functions in both metabolism and biosynthesis. And as we said, we have certain exceptions: amino acid that does not undergo transamination, and that include lysine, threonine, proline, and hydroxyproline. And this is the process of transamination, where this alpha amino acid will be converted to it is alpha keto acid. Counterpart here, where this alpha keto acid will take up the amino group to form a new amino acid. The transaminase, of course, uses pyridoxal phosphate, the active form of vitamin B6, as a coenzyme. Transamination of non-essential amino acids synthesized in this way, the alanines, aspartate, glutamate, and glutamate, and serine. Transamination of pyruvate, this the alpha, the carbon skeleton here, will take the amino group either from glutamate or aspartate by a transaminase to form alanine, and one-step reaction. Likewise, oxaloacetate will form aspartate reversibly, and glutamate definitely, yeah, in addition to being synthesized by transamination, uh, glutamate can be synthesized by a reductive amination of alpha-ketoglutarate by the glutamate dehydrogenase. We have already also discussed it. Glutamate can be synthesized from proline and from glutamine by glutamate synthetase, particularly in bacteria. And this is transamination of the carbon skeletons alpha ketoglutarate. Alanine here is the source of the amino group to form glutamate, while alanine will be converted its carbon skeleton is pyruvate by alanine transaminase. Glutamate, as we said, can be used by reductive amination, that is alpha ketoglutarate undergoes reduction by glutamate dehydrogenase, where NADPH is a source of hydrogen, and amination, there is addition of an amino group to form glutamate and water and NADP. From glycolytic intermediate, D3-phosphoglycerate, we can synthesize serine by oxidation and then transaminated to phosphoserine and then dephosphorylated to form serine. Another way of synthesizing serine is from glycine by the action of serine hydroxymethyl transferase in reversible reaction. This is the first reaction whereby serine is synthesized from the intermediate phosphoglycerate using an NADH as a coenzyme, formation of phosphohydroxypyruvate, and then amination, an amino group is donated by this alpha amino acid to form alpha keto acid. This amino group now is added to the phosphohydroxypyruvate to form phosphoserine, and then as there is hydrolysis and phosphate here is released as inorganic phosphate, and serine is finally formed. Serine can be synthesized from glycine in one-step reversible reaction by serine hydroxymethyl transferase, which transfers a hydroxymethyl group between the two amino acids. So we can synthesize serine from glycine and vice versa. As we said, in addition to transamination and synthesis from the precursor molecules, the body can assimilate free ammonia, and that is to say, it will take up NH4 ammonia compound and incorporate it into an amino acid to form a new amino acid. And our examples here, of course, are glutamine and asparagine. Glutamate via glutamine synthase will assimilate this free ammonia into the glutamate to form the second amino group, glutamine. And likewise, asparagine by asparagine synthetase will use amino group, will incorporate free ammonia to form asparagine. And here, to compare and contrast glutamate dehydrogenase and glutamine synthase, both of them synthesize or act on the amino acid glutamate and glutamine. Glutamate dehydrogenase fixes inorganic nitrogen, assimilate free inorganic nitrogen, while glutamine synthase does the same thing. However, glutamate dehydrogenase will fix inorganic nitrogen to form an amino linkage, while glutamine synthetase will form an amide linkage using the same source of nitrogen. And both of them are coupled to highly exergenic reactions. It is the conversion of NADPH or NADH to NAD or NADP in glutamate dehydrogenase, and it is phosphorylations of ATP to ADP, an inorganic phosphate, in the case of glutamine synthesis. The third way of synthesizing non-essential amino acid, as we said, is through modification of an existing amino acid. And our examples here are cysteine, glycine, tyrosine, and proline. Cysteine is synthesized by donating two atoms from two different amino acids. One of them is essential, and that is methionine, and the other one is non-essential, which is serine. So the body will use two existing amino acids, methionine and serine, to synthesize cysteine. From methionine, ATP is hydrolyzed to form acetylnosyl methionine. So this adenosine moiety here comes from ATP. We need an acceptor for methyl group, so a methyl group will be removed from methionine to form acetylnosyl L-homocysteine, and then this compound is hydrolyzed with release of adenosine to form L-homocysteine, and then L-homocysteine will be further hydrolyzed to cystathionine, which will be rehydrated back to yield two compounds, cysteine and homoserine. Biosynthesis of glycine can occur by one of four ways. Number one is from serine, as we said, by serine hydroxymethyl transferase. Glycine can be synthesized from glycoxylate by glycine transaminase, which uses glutamate or alanine as a source of amino group. And this reaction here is reversible, but it favors direction of formation of glycine. The third way for synthesis of glycine is from carbon dioxide and ammonia and one carbon unit by glycine synthase, and the fourth way is from proline. We are going to discuss here one example, which is synthesis from serine by serine hydroxymethyl transferase. As you can see, the reaction is reversible, but generally prefers or preferential goes in the direction of the formation of glycine. Biosynthesis of tyrosine by hydroxylation of phenylalanine. So whenever you have phenylalanine in the body can use this phenylalanine to synthesize tyrosine, but the reverse does not occur. So tyrosine cannot replace phenylalanine. Synthesis of tyrosine from phenylalanine requires the action of the enzyme phenylalanine hydroxylase. It is a mixed function oxygenase. It introduces one oxygen into phenylalanine into the para position here to form this hydroxyl group. So it will convert phenylalanine to tyrosine, and at the same time it reduces molecular oxygen into water. This is the action of a phenylalanine hydroxylase. This reaction requires a coenzyme tetrahydrobiopterin, which will be reduced to oxidized to dihydropterin, and then dihydropterin reductase will regenerate the tetrahydropterin again using NADPH as a source of hydrogen. Biosynthesis of proline is by reduction and cyclization of glutamate, or through the reverse of reaction of proline synthesis. So we use glutamate to synthesize proline. We need two steps. Glutamate will first be dehydrogenated and oxidized to proline dehydrogenase with release of water to L-glutamate gamma semialdehyde, and then non-enzymatically water will be released to form delta-2-pyrrolidine 5-carboxylate, and then by glutamate semialdehyde dehydrogenase using NADH. There is cyclization to proline. And this ends our discussion of biosynthesis of non-essential amino acids. Thank you very much.